Hello, and welcome to the 5th International Symposium on Sigma-2 Receptors, Role in Health and Disease. My name is Wayne Bowen, and I'm going to be talking to you today about some of the work we've done with Sigma-2 receptor ligand-induced programmed cell death mechanisms in triple-negative breast cancer cells. Now, triple-negative breast cancer is a particularly aggressive form of cancer characterized by a lack of expression of the traditional targets for hormonal drug therapy, namely the estrogen receptor, the progesterone receptor, and human epidermal growth factor receptor type 2 upregulation. And these are higher grade tumors at diagnosis with poorly differentiated cells. The disease is more prevalent in premenopausal women and women of African descent, and it makes up about 15 to 20% of all breast cancers. Now, this paucity of therapeutic targets leaves chemotherapy as the standard of care and treatment of TNBC, and this has a high rate of relapse. And so there's an unmet need for the development of targeted therapies for this disease. Now, TNBC is a heterogeneous disease. It shares some of the characteristics of basal-like breast cancers, as indicated by this Venn diagram. And it's been subdivided into four general subclasses, namely the basal-like 1, which is characterized by an elevation of cell cycle and DNA damage genes. Basal-like 2, which is enriched in growth factor signaling and epithelial markers. Mesenchymal, which has elevated expression of genes involved in EMT and growth factor pathways. And then luminal androgen receptor, which is characterized by upregulation of genes responsive to the androgen receptor. And it's hoped that this type of molecular characterization of TNBC will lead to the development of targeted therapies that would aid in individualized treatment of the disease. Now, in the studies that I'm going to be talking to you about today, we've used 3 cell lines which are representative of 3 of the subclasses of TNBC. The MDA-MB231 cells, which is representative of the mesenchymal type, which is enriched in pathways involved in cell motility and cell differentiation, and which has mutations such as those shown here, such as BRAF and mutations in KRAS. The 453 cells, which are representative of the luminal androgen receptor type, which is heavily enriched in hormonally regulated pathways around steroid synthesis, porphyrin metabolism, and androgen and estrogen metabolism, and characterized by mutations in genes such as PI3K and PTEN. And then the 468 cells, which are representative of the basal type 1, which is heavily enriched in cell cycle division genes and pathways and cell cycle checkpoint loss, and is characterized by mutations in PTEN, P53, and so forth. Now, the first thing we did was we looked at these cells to see if they express sigma-2 receptors, and we used treated DTG in presence of plus pentazocine to mask the sigma-1 receptor, and found that all 3 subtypes express sigma-2 receptors, with the Bmax values shown here and the KD values for treated DTG shown here. Now, what was surprising, however, was that when we compared these cells to the less aggressive MCF7 cells, we found that these TNBC cells have lower levels of sigma-2 expression than the less aggressive MCF7. And this is surprising because it's known that rapidly proliferating and more aggressive cells tend to have higher levels of upregulation of sigma-2 receptors. So this was sort of a surprising finding. However, on the other hand, normal human mammary epithelial cells, these HMEC cells, had little to no detectable sigma-2 receptor expression based on treated DTG binding. So the next thing we did was we looked at 2 sigma-2 receptor ligands, the structures of which are shown here, for their effects on these cells. The first ligand we looked at was MAM03055A, which is a compound developed in collaboration with Chris McCurdy and his group. And this compound has some interesting characteristics. First of all, it is a bivalent, a homobivalent compound. And it also binds pseudo-irreversibly to the sigma-2 receptor. And its binding characteristics for sigma-1 and sigma-2 are shown here. We also looked at SV119, which was developed in the laboratory of Bob Mack, and he was kind enough to provide us with this compound. Its sigma-2 and sigma-1 binding characteristics are shown here. And you can see that both compounds have good affinity for sigma-2 and good selectivity for sigma-2 over sigma-1. Now we looked at the effect of these compounds on cell viability, and both ligands induce dose-dependent cell death in all of the breast cancer cell lines. And so what we have here is cell viability expresses percent cytotoxicity as a function of dose. And you can see that the EC50 values shown here, all three TNBC cell lines are killed by the compounds. And what you notice here is that the MAM compound is more potent than SV119. And this is despite the fact that SV119 has a higher sigma-2 binding affinity. Now we know that the lipophilicity of compounds affects their potency, so it could be that SV119 is less lipophilic and has more difficulty crossing the cell membrane. But there could be other factors as well that I'll talk about later in the talk. Another thing to notice here is that the MCF7 cells, the less aggressive MCF7 cells, has an EC50 that is comparable to the TNBC cells. And this suggests that, well, this shows that the factors that lead to the aggressiveness of the TNBC cell lines is overcome by the sigma-2 receptor ligands. And so the sigma-2 receptor ligands are just as easily kill the TNBC cells as they kill the less aggressive MCF7 cells. On the other hand, the normal cells, the human mammary epithelial cells, which had much lower, little to no expression of sigma-2 receptors, are much less sensitive to the compounds. And so this is consistent with the lower level of sigma-2 receptor expression, and shows that there is a good therapeutic window for these types of compounds. Now, what we did next was we looked at several cell death pathways that had been shown previously in other cell types to be involved in the mechanisms of cell death. And so we looked at mitochondrial depolarization, which we had shown previously is a component of cell death as we characterize it in SK and SH neuroblastoma cells. And what we found using the JC1 ratiometric assay to monitor mitochondrial membrane potential, and using valinomycin as a positive control, the potassium ionophore, what we were able to show is that in all three cell lines, both sigma-2 receptor ligands induce a dose-dependent loss in mitochondrial membrane potential. So this appears to be at least contributing to the mode of cell death in these cells. Now, since there is mitochondrial depolarization, as you know, mitochondrial depolarization results in the release of apoptogenic factors from the mitochondria. And in this case, cytochrome C can be released, and cytochrome C can activate caspases downstream, particularly caspase 3. So we looked at caspase activation, which has been shown in other cells to be a component of cell death mechanisms in other cell types. And what we were able to show was that in all three cell types, both ligands induce a dose-dependent increase in caspase 3 activity using a fluorescent assay. And you see here in all three cases, there's this dose-dependent increase in caspase-3 activation. However, the ability of caspase-3 to be activated by these sigma-2 receptor ligands is uneven across these cell lines. So as you can see, the 231 cells are much more sensitive, if you look at the doses here, compared to the other cell lines, and the 453 cells are the least sensitive. So caspase-3 activation does not appear to be even across these cell lines. So we look next at PARP. PARP is cleaved by caspase-3. It's a major substrate of caspase-3. PARP cleavage results in a decrease in the ability to do DNA repair, leading to apoptosis. So we looked at PARP cleavage across these three cell lines. And we did this by Western blot, looking at the intact PARP and then its fragment, the 89 kilodalton fragment, over 3 time points, 12, 24, and 36 hours, using a 30 micromolar dose of each ligand. And what we were able to demonstrate is that there is a time-dependent increase in PARP cleavage, as shown here. The black bar is the intact PARP, and the white bar is cleaved PARP. So over time, MAM produces this time-dependent increase in PARP cleavage, and we see the same thing with the SV119 compound in the 231 cells. In the 453 cells, we see a similar thing where we have this time-dependent increase in PARP cleavage with MAM, except with the 453 cells with MAM, we see this sort of bell-shaped relationship in cleaved PARP, where there's more PARP cleaved at the earlier time points and at the later time points. And we don't know what this means. It may have something to do with the irreversible binding nature of the compound. And we see the same, we see a similar profile with SV119, although we don't see much of this sort of bell-shaped relationship with SV119. And then in the 468 cells, again, we see this time-dependent increase in PARP cleavage with both MAM and with SV119. So PARP cleavage also appears to be contributing to the cell death mechanisms in these cells. Now, because PARP is cleaved by caspase, and caspases are activated, and we only measured caspase-3, but there could be other caspases that are also activated. We wanted to address whether activated caspases are essential for this mode of cell death. So we looked at the effect of caspase inhibitors, of a caspase inhibitor on the action of these sigma-2 receptor ligands. And so using ZVAG-FMK, which is a broad spectrum caspase inhibitor, what we were able to show is that in all 3 cell types, using MAM, and the gray bar is MAM alone, and the white bar is MAM in the presence of 100 micromolar ZVAD. What you see in all 3 cell types is that ZVAD is unable to protect the cells from the cytotoxicity induced by various doses of the MAM. So MAM-induced cell death appears to be caspase independent. And we see the same thing with the SV119 compound. In the 3 cell lines, in the 3 cell lines, ZVAD-FMK is unable to alter the cytotoxic potency of either of the sigma-2 receptor ligands. So the mode of cell death appears to be caspase independent, or caspase activation isn't essential for the cell death. There's also evidence that autophagy plays a role in sigma-2 ligand-induced cell death. So we also looked at autophagy in these 3 cell lines. And for autophagy, we looked at 2 markers of autophagic signaling. We looked at the conversion of LC3B1 to LC3B2, looking at this by Western blot. And we also looked at P62, which is another marker of autophagy. And what we see here, again, looking at 3 time points with a fixed dose of 30 micromolar of each of the ligands. For MAM, we see this dose-dependent increase in the formation of LCB32. The black bar is LC3B1. The white bar here is LC3B2. And you see that there's a time-dependent increase in the formation of LC3B2, and also a time-dependent increase in the accumulation of P62, the other marker of autophagy. Similar thing was observed with LCB, with SV119, where we see this time-dependent increase in the formation of 2, and also a time-dependent increase in the accumulation of P62, although in these cells, it's rather latent. We don't see significant accumulation of P62 until 36 hours. In the 453 cells, again, we see a time-dependent increase in the formation of LCB32 with MAM. And with respect to the P62 with MAM, we see, again, this sort of interesting bell-shaped relationship for a downstream signaling pathway. Again, we don't know what this means. Again, it could have something to do with the irreversible nature of the compound. And then over here with SV119, we see, again, the time-dependent increase in the formation of LCB32, and again, a time-dependent increase in the formation of P62. And then finally, in the 468 cells, again, the same observation. MAM induces a time-dependent increase in 2, time-dependent increase in the formation, accumulation of P62, and SV119, we see the same sort of thing. Time-dependent increase in formation of LC3B2, and a time-dependent increase in the formation of P62. So in summary, TNBC cells representing various disease types express sigma-2 receptors. Normal breast cells express little or no sigma-2 receptor binding activity as detected by tritiated DTG binding. The sigma-2 receptor ligands induce dose-dependent cell death in TNBC cells. And each ligand has a similar potency across the TNBC cell types, despite the intrinsic genomic differences. And this indicates that these genomic differences that lead to increased proliferation and drug resistance and so forth do not affect the potency of the sigma-2 receptor ligand. The potency is comparable to that of the less aggressive MCF7 cells. So again, the factors that lead to the aggressiveness of the TNBC cells is overcome by the sigma-2 receptor ligands. And normal breast cells are more resistant to sigma-2 ligand-induced cell death, and this may have something to do with their very low expression of sigma-2 receptors. Structurally diverse sigma-2 receptor ligands activate the same multiple programmed cell death pathways. They both induce mitochondrial depolarization, caspase activation, although caspase activation does not appear to be essential for cell death, PARP1 cleavage, and autophagic pathways. Now, the fact that structurally diverse sigma-2 receptor ligands activate the same downstream cell death programs would suggest that they are interacting through the same target. However, we have yet to verify that these compounds are acting through sigma-2 receptor TMEM97. We have yet to do knockout experiments, for example, to show that this protein is in fact involved. There is evidence from Bob Mack's lab that you can knock out TMEM97 without affecting the cytotoxic potency of certain compounds. So we really need to verify that TMEM97 is the actual target. The role of PGR-MC1, does it play a role here? PGR-MC1 is known to form a functional complex with TMEM97. So is there a role for PGR-MC1 here? Are other signaling partners potentially involved? Does TMEM97, for example, combined with other signaling partners to produce different downstream effects. And then finally, are there other proteins with sigma-2-like pharmacology that may be involved? Maybe TMEM97 is not the primary target of these ligands. It could be that there are other proteins that have sigma-2-like pharmacology. We know that there's evidence that there are other proteins that bind sigma-2 receptor ligands, but which have low affinity for tritiated DTG, for example. So we may not be detecting these proteins. And so I've been careful throughout the talk to talk about sigma-2 receptor ligand-induced effects because we actually have yet to actually verify what the target is mediating these effects. So this is work that needs to be done in the future. So there's much more work that needs to be done here. So let me acknowledge those that have that actually did the work: Sherry Liu and Hillary Nicholson, two former graduate students in the lab; Bridget McVeigh, a former master's student in the lab; Neelan Wong, also a master's student in the lab; and Blessing Akabundu, who is a rotation student who came through the lab. Our collaborators, Chris McCurdy and Marco Montanelli, who were involved in the development of the MAM compound. And then Bob Mack for his generous gift of the SV119 compound. Thank you very much.